Hello everybody, myself Dr. Nupur Sadda, Botany Faculty Resonance Pre-Medical Division. Today we are here to discuss the AIIMS 2019 paper which is held on 25th May in the morning session. So first let me tell you these are purely memory based questions and their solutions. If you look at the botany section, let us see question number 1, it is related to the diagrams. Okay. If we see figure A, this is of fucus, that is of brown algae. B, that is of porphyra, that belongs to red algae. C, that is of laminaria, that belongs to brown algae. And the last one, D, that is of polysiphonia, that is red algae. So according to it, the correct answer will be 3. Question number 2, select the correct diagram. Here basically the diagram of female gametophyte is given or the embryo sac is given. If we look at it, it is a 7-celled 8-nucleated structure. That is, it consists of the antipodal cells. These are the 3 antipodal cells. 2 polar nuclei are there, and this is the egg apparatus which consists of the 2 synergids and 1 egg cell. So here the correct option will be 1. Second, third and fourth will not be correct. Why? Because here only 2 antipodal cells are there, and if we look at this diagram, here only 1 of the polar nuclei is there, and if we look at the fourth figure, here the egg cell is missing. So the correct answer will be first. Let us see question number 3. This is describing basically the process of transcription in case of prokaryotes. Transcription is the synthesis of messenger RNA from the DNA. Okay, it consists of 3 steps, that is the initiation, elongation and termination. But when we talk about the prokaryotes, here RNA polymerase itself is capable only of the process of elongation. For the process of initiation, it will require the sigma factor, and for the process of termination, it will require the rho factor. So we can say there are 3 steps: initiation, elongation and termination. Initiation with sigma factor and RNA polymerase, elongation only RNA polymerase, and termination RNA polymerase and rho factor. So here they have mismatched the sequence. If we look at it, this is referring to the initiation, this is referring to the termination, and C is referring to the elongation. So here the correct answer will be 2. Let us see question number 4. It is match the columns regarding the different types of interactions which we have studied in the chapter of ecology, organism and population. If we talk about parasitism, parasitism is a plus minus type of relationship in which one of the organism is benefited while the other is harmed. Commensalism, commensalism is a type of relation in which one of the organism is benefited while the other remains unaffected. So plus 0 type of relationship. If we talk about the C, that is amensalism. Amensalism is the interaction in which one is harmed while the other remain unaffected. So it will correlate with 4. And D, mutualism. Mutualism is the interaction in which both the organisms are benefited from each other. So it will be plus plus. So here the correct option is 4. Let us see question number 5. Genes on the same chromosome can be, first is linked, second is homologous, third is autosomes, and fourth is identical alleles. Here the correct answer will be linked genes. The genes which are present on the same chromosome, they can be linked. Homologous and autosomes, here they are talking about the chromosomes. Identical alleles actually में जो alleles होते हैं, they are present on the same loci on the homologous chromosomes. Same loci means this one, this, okay, not on the same chromosome. So here the correct option is first. Let us see question number 6. It is again match the column type of question which has been asked from the chapter plant breeding. It is a direct question. Here the crop type or the variety is given, and in the second column the diseases have been given to which these varieties are resistant. Okay. So let us match it. Pusa shubra, basically it is the variety of cauliflower. Pusa swarnim, if we talk about, this is a variety of mustard which is resistant to white rust of crucifers. Pusa sadabaha, this is the variety of chili which is resistant to chili mosaic virus. And fourth, himgiri, this is the variety of wheat which is resistant to leaf and stripe rust. So according to this, the correct option will be second. Let us see question number 7, which of the following is correct about the symplast. Now this is the term which is taken from the chapter, that is transport in plants. There are two types of pathways, that is symplast and apoplast. If we talk about the symplast, it is the living connection, that is a connection, cytoplasmic connection which occurs by means of the plasmodesmata. Whereas if we talk about the apoplast, that refers to the non-living connection which includes the cell wall and the intercellular spaces. So here if we go through the options, first is living continuum, second is cell wall and intercellular space, third is non-living continuum, and fourth is none of these. So here the correct answer will be 1. Let us see question number 8. This is match the column type of question which has been asked from cell biology. Different types of plastids are given and we have to match it along with the corresponding function. So let us see that elaioplast. Elaio refers to the fat. This is related to the storage of fat. Second, eluroplast. Eluro is related to protein. So this is related to the storage of protein. Third is amyloplast. Amylo refers to the starch. So here amyloplast refers to the storage of starch, and the last one is chromoplast. This basically consists of the fat soluble pigments, that is carotenoids. Okay, so colored pigments, this will correlate with D. So here the correct option is 4th. Let us see question number 9. Virus-free plants can be formed by. Here the options given here are meristem culture, second is callus culture, third is somatic cell culture, and fourth is the protoplast fusion. Now let us know what are these basically related to. Meristem culture, if we talk about, this is the culture of the meristem. Okay, so virus infected plant, if we take the apical meristem or the axillary meristem from it and culture it, then we will get the virus free plant. Why? Because the meristem is related to the active cell division which is free from the virus. So here the correct answer will be first. Question number 10, which of the following is correct set of macronutrients? Here different options have been given and we have to choose the set of macronutrients. Macronutrients are basically CHONS, P, K, Mg. So according to it, the correct option is 4th. Question number 11, percentage of G plus C is. According to the Chagaf's rule, if we talk about, the total amount of purine is equal to the total amount of the pyrimidines, or we can say the molar amount of the adenine is equal to that of thymine and the molar amount of guanine is equal to that of cytosine. So according to this concept, the correct answer will be first. Question number 12, arrange them on the basis of the increasing size. Here 4 terms have been given, that is the nucleotide, chromosome, gene and genome. Firstly, you should know what is the genome. Genome is the complete set of information of an organism. Okay? So, and this genome is a collection of chromosomes we can say. ठीक है? और chromosomes पे क्या present होते हैं? The genes are present on the chromosomes. And if we talk about the nucleotide, it is a building block of the DNA. So according to this, the correct option will be 4. Question number 13, which of the following is a microelement? It is a direct question from mineral nutrition. Here the correct answer will be 3. Manganese. Question number 14, the genetic code of proline R. It is a direct question from molecular basis of inheritance. First is CCC, CCG and CCU. This is the correct answer. So this is option 1. Question number 15, the coding strand of DNA is given in 5-3- direction and the sequence of the messenger RNA has been asked. If we looked at the transcriptional unit, basically there are the 2 strands. First is the template strand or the non-coding strand which runs in 3-5- direction, and the second one is the coding strand that runs in 5-3- direction. So here the sequence of messenger RNA has been asked and the coding strand sequence is given, means the sequence of this strand is given. If we talk about the messenger RNA, the sequence will be same as that of the coding strand, but instead of T, that is thymine, uracil will be present because it is a messenger RNA. So, if we look at the options, but the direction will remain the same, that is 5 dash, 3 dash. So, here if we go through the option, the correct option is second. Question number 16, match the following. Here the different shapes or diagrams have been given and we have to identify it. If we look at the first one, this is the coccus shape of bacteria, this is the bacillus, and this is the diagram of the bacteriophage. So, here the correct option will be second. Let us see question number 17, synthesis of lipid is a function of. First option is SER, that is smooth endoplasmic reticulum. Second is RER, that is rough endoplasmic reticulum. Third is Golgi body, and fourth one is mitochondria. As we know that smooth endoplasmic reticulum is basically related to the synthesis of the lipids, steroids, and also involved in the detoxification of the drugs. If we talk about RER, it is involved in the synthesis of proteins and the ribosomes. So, here the correct option will be first. Question number 18, DNA polymerase links nucleotide by forming which type of bond? First is the phosphodiester bond, second is hydrogen bond, third is glycosidic bond, and fourth is the ester bond. If we talk about DNA, it is a deoxyribonucleic acid which is a polynucleotide. Okay, polynucleotide है तो बहुत सारे nucleotides जो हैं वो मिलकर polynucleotide chain बनाएंगे, and these nucleotides they are connected by means of the phosphodiester bond. So here the correct option is first. Question number 19, match the following. Here the different types of fruits have been given and their examples have been given. We have to match them. First is siliqua. Silica का जो correct example है that is mustard, brassica. Caryopsis, इसका जो correct set of example आएगा that is triticum. Berry, first one that is Lycopersicum esculentum, that is a botanical name of tomato, and Cypsela, third that is Helianthus anus, sunflower. So here the correct answer will be third. Question number 20, select the wrong statement. Let us go through the options. First option is the human genome contains 3164.7 million nucleotide bases. Second is less than 10% of the genome codes for protein. Third is repeated sequences make up very large portion of the human genome. And fourth is chromosome 1 has most genes, that is 2968, and Y has the fewest, that is 231. Okay, so this is a direct NCERT based question which has been taken from the chapter that is molecular basis of inheritance from the human genome project. So if we go through these options, the incorrect one is second, because here 2% of the genome codes for protein, less than 2%. So the correct answer will be second. Question number 21, homologous chromosome can be defined as. First is chromatids of the same chromosome. No, this is incorrect. Let us see the second option, same chromosome, same gene, different allele and different order. Here different order will be incorrect. Why? Because if we consider the homologous chromosomes, basically they are the zygotic pair of chromosomes which pair up during the meiosis, zygotine stage, that is we also call it as synapses. Okay? One is a paternal and one is the maternal chromosomes. If we consider the genes, the different genes will be present on the same chromosome, and if we talk about the homologous pair पे जो मिलेंगे different alleles भी मिल सकते हैं इस केस में, सिमिलर एलील्स भी मिल सकते हैं जैसे कि इस केस में, लेकिन इनका जो सीक्वेंस रहेगा वो सेम रहेगा. ठीक है, तो यहाँ पर डिफरेंट ऑर्डर वाला हो गया, इसलिए दिस विल बी इनकरेक्ट. इफ यू लुक एट द थर्ड ऑप्शन दैट इज सेम क्रोमोसोम, डिफरेंट जीन, सेम एलील. डिफरेंट जीन सेम एलील, तो यहाँ पर सेम एलील वाला जो कंसेप्ट है वो क्या हो गया, इनकरेक्ट. इफ यू गो टू द नेक्स्ट ऑप्शन दैट इज सेम क्रोमोसोम, सेम जीन, different allele in the same order. This is the correct one. So here the correct answer is 4. Let us see question number 22, match the columns again. Pleiotropic gene is the gene which shows multiple phenotypic expression. Okay, so here if we match it, this is the correct one. B, codominance. In case of codominance, both the alleles, they express themselves equally. So in this case, if we see, the first one is matching. Epistasis. Epistasis is a type of non-allelic interaction in which one gene masks the effect of the other non-allelic gene. So we can say fourth one. And D, mutation. It is the sudden change in the genetic makeup of an organism, or you can say change in the nucleotide. So according to this, the correct option will be 3. Let us see question number 23. Again, match the column is there. K.C. Mehta, he proposed the rust cycle related to the rust fungi Puccinia. Maheshwari gave the concept of haploid culture, or anther culture, we can also call it as a pollen culture. Cohen and Boyer is related to the first recombinant plasmid, and the last one, that is Singer and Nicholson, proposed the fluid mosaic model. So according to this, the correct answer will be second. Let us see question number 24. Choose the correct statement. If we look at the first option, transcription and translation occur in same compartment for prokaryotes. As we know, as in the case of prokaryotes, unorganized nucleus is present. So both the processes, that is transcription and translation, will occur in the cytoplasm itself. They are coupled. So this is the correct statement. Second one, monocistronic RNA, more than one structural genes under the single promoter. This is wrong here. Single, अपन कहेंगे structural gene is under the single promoter. Then third one, introns and exons both code for the protein synthesis. No, this is wrong. Introns are basically the non-coding sequences and exons are the coding sequences. If we look at the fourth option, in prokaryotes, splicing and tailing occurs before translation. This is wrong. Why? Because in case of prokaryotes, splicing and tailing does not occur. It is related to the processing of the RNA, and it occurs in case of the eukaryotes, not prokaryotes. So here the correct answer is first. Let us see question number 25. It is assertion and reasoning type of question. Assertion, in eukaryotes, both intron and exon are transcribed to form the heterogeneous nuclear RNA. This is correct because in case of eukaryotes, the split gene concept is present. Split gene means it consists of coding as well as non-coding sequences. Later on, this introns or the non-coding sequences, they are removed by the process of splicing. Okay, so this is correct. If we go to the reason, that is splicing is required in case of prokaryotes, this is incorrect because in case of prokaryotes, splicing or the processing of the messenger RNA does not take place. So here the correct option will be 3. If we go to the question number 26, assertion parthenocarpy involves the formation of seedless fruit. This is correct. If we go to the reason, that is apomixis occurs without fertilization. Apomixis is the production of seed without fertilization. It has got importance in the production of hybrid seeds. So it is a type of asexual reproduction which mimics sexual reproduction. Here both the assertion as well as reasoning is correct, but reason is not the correct explanation of the assertion. So here the correct option will be second. Let us see question number 27. Here the assertion given is soma clonal variation produce slight differences in the plant. If we talk about the soma clones, exactly they are the copies, similar copies which are produced during the process of tissue culture. That's why we call it as the soma clones. But sometimes there may arise variation due to the process of mutation. If we look at the reason, they are produced while performing the tissue culture. This statement is correct, but it is not able to explain the assertion. So here the correct answer will be second. Now let us see question number 28. Here the assertion given is in eukaryotes, transcription occurs in the nucleus. If we talk about the eukaryotes, in case of eukaryotes, organized nucleus is present. That is, the process of transcription will be carried out in the nucleus and the process of translation will be carried out in the cytoplasm. But if we talk about the prokaryotes or the bacteria, here the process of transcription as well as translation, that is carried out in the cytoplasm because it has unorganized nucleus. So if we look at the reason here, in bacteria, transcription and translation occurs in the cytoplasm. Both our statements are correct, but the second one is not the correct explanation of the assertion. So here the correct answer will be second. Let us see question number 29. Here the assertion given is fig and wasp cannot complete their life cycle without each other. That's true. Fig provides the oviposition site as well as the ovules to the growing larva of the wasp, and wasp helps in the pollination of the fig. Reason, it is a mutualistic relationship, that's true. It is because it is an obligatory mutualistic relationship between the two. So, here the correct answer will be first. Now, look at question number 30, that is assertion biofortification is used to increase the nutrient value of the crop. This is correct. If we look at the reason, meristem culture is used to obtain the virus resistant plant. Meristem culture is generally used to obtain the virus free plants, because meristem itself is a part which shows active cell division, which is generally free from virus. So, here the correct option will be 3, as this part is wrong. Let us see question number 31. Here the assertion given is isolated protoplasts are used for somatic hybridization. Firstly, we should know what is somatic hybridization. It is the process of formation of the somatic hybrid, in which the two somatic cells are brought close to each other. Firstly, their cell wall is removed, and the cell without cell wall, that is known as a protoplast. And then their hybridization is brought about by either chemicals or electrically, that is known as electrochemical fusion or electrical fusion or chemical fusion. So, that results in the formation of the somatic hybrid. So, this is correct. If we look at the reason, callus culture does not allow variation. This statement is true, but it is not explaining the assertion. So, here the correct answer will be second. Now, let us see question number 32. In C3 cycle, the first stable compound is 3-carbon compound. That is true. That is why we call it as the C3 cycle or the Calvin cycle, because here the first stable product formed is the 3-phosphoglyceric acid, or we can also call it as 3-phosphoglycerate. Look at the reason. In C4 plants, Calvin cycle is absent. C4 plants are also showing, we can say, Hatch and Slack pathway. Here Cran's anatomy is present and dimorphic chloroplasts are there, because here 2 times carboxylation is carried out. One is in the bundle sheath cell and the second one is in the mesophyll cells. So, here we can say that Calvin cycle is not absent. So, here the correct answer will be 3. Let us see question number 33. Assertion given here is in vernalization, flowering is induced by low temperature. That is true. Vernalization is the process of induction of the flower by low temperature treatment. Reason given here is abscisic acid is growth inhibiting hormone. The statement is correct, but it is not explaining the assertion given here. So, here the correct answer is second. If we look at question number 34, assertion given here is in commensalism, one organism is benefited and other is unaffected. This is true. It is the defining property of the commensalism interaction. If we look at the reason, cattle aggregate bird, and cattle is an example of commensalism. It is just an example. It is not explaining the assertion. So, here the correct answer will be second.